also the Melonia in manufacturing awardees. And very impressively, she was selected as one of the 50 called Inspiring 50 Italy in 2021. Her research interests include biomedical translation, instrumentation development, and image processing for unbiased assessment of tissue property. She's the expert in OCT, optical coherence tomography, has very broad application in biomedical imaging, even has some relevance with cultural heritage, material processing. And she will talk about high definition, non-invasive 3D imaging with OCX. And just a little bit side, I have a business travel very soon, so need to go to the airport. So I may leave early, just let you know. And let's welcome Dr. Canavesi for her talk.
Thank you. Thank you so much for the introduction and for the invitation to be here. I was joking that my office is like 5 minutes away, but it's rare that I come over, so it's great to meet everyone.
It's great to have you.
Thanks. So I think most of you have already heard kind of my background, but as I started Light Optech when I was finishing my PhD at the University of Rochester back in 2013. And the seed that started that brought us to start the company was a program by the National Science Foundation that's called I-Core that really gave us the foundation of learning how to go about commercializing a highly technical invention. And a few years later, in 2018, we also took part in the Illuminate Accelerator here in town, which is the only optics and photonics accelerator run by NextCore downtown in the Sibley building. Obviously, Rochester has a lot of optics foundations right here. We've been funded primarily through small business innovation research grants, both from the National Science Foundation and the National Institute of Health. And we've also received funding from New York State through local programs through the CIS and COE institutes. So the motivation, the outline of the talk is I'll talk about the motivation for all of this technological development, which is optical biopsy, and using that to guide surgery and other interventions that impact patient health. And we'll talk about the core technology using Gabor domain optical coherence microscopy, which is a high lateral resolution variant of optical coherence tomography, OCT, and combining GDOCM and OCT in a novel dual modality instrument called OCX. We'll discuss, as Dr. Chow mentioned, there's numerous applications in the biomedical space. I'll talk about a few of those, including cervix, skin, and cornea. So the motivation really is to be able to resolve microscopic structures down to the cellular level inside of living tissue in a completely non-invasive manner. And the requirements for this application are to be able to resolve down to the micron scale in 3D and inside of tissue at clinically relevant depths. So at least a millimeter inside of tissue. And we want to be able to do this over a large field of view, relatively large, compared to microscopy. So over a millimeter field of view, and in a way that's non-invasive, so that's a true optical biopsy without any cutting. So to put this into perspective, there's this chart shows different imaging modalities in terms of two key parameters. So on the vertical axis, there's lateral resolution, so how small of a feature you can resolve, and the horizontal axis is the depth of penetration inside of the sample. And as you can see, they all kind of fall along this diagonal because there's a trade-off between these parameters. And so we start from these with the goal of imaging with this 1 micron resolution. So there's a set of these technologies that have insufficient resolution to be able to get down to the cellular level, such as ultrasound. So ultrasound can penetrate through the entire body. This is an example of an ultrasound image of the cornea, so the outermost part of the eye, and it has excellent penetration, depth penetration, but lacks in the lateral resolution. So OCT, if you're not familiar, optical coherence tomography is kind of the equivalent of ultrasound, but using optical wavelengths instead of sound. So you send a wave through tissue and detect what's reflected back. But with ultrasound, you basically measure the time of flight of when the beam is reflected back. With light, there's some additional considerations that need to be done to be able to make that measurements that we'll talk about in a little bit that involve interferometry to be able to make that time of flight measurement. But that's the same principle. So you're using light instead of sound. So the penetration is not as much as ultrasound, but you get an improvement in the lateral resolution. So what you see there is a cross-sectional image with OCT, which is a typical presentation of OCT images. It has micron scale depth resolution capability, so you can see very well layers, but it still lacks the lateral resolution capability. So to really break into the cellular space, you have to go to microscopy. So there's confocal microscopy, as shown here, imaging the endothelial cell layer. So you can make beautiful resolution of the borders of the cells down to micron and submicron level, but really at the expense of penetration along the depth. And also in reduction in field of view. So typically, confocal microscopy field of view is less than 0.5 millimeter. And then we have so a variant of optical coherence tomography that was introduced to achieve cellular resolution. It's called optical coherence microscopy, and it achieves micron scale resolution, but only over a very limited depth of focus. So here is some example of the endothelial cells that you can see with OCM. Now, with the Gabor domain optical coherence microscopy, we kind of bridge the capabilities of OCT and OCM, having both the depth resolution, so the depth of focus is recovered through the entire depth of the sample, and we have micron scale lateral resolution. So you can see on the bottom the endothelial cells are beautifully resolved, and over a field of view that's greater than 1 millimeter, and we achieve that micron scale resolution in 3D. And what we've done is combine OCT and GDOCM in a single instrument that we've called OCX, because it has the dual modality capability of OCT over a larger field of view up to 10 millimeter, and then the GDOCM up to 2 or 3 millimeter field of view with a micron scale resolution. So let's talk a little bit more about the two different pieces, OCT and GDOCM. So the concept of OCT is shown here in what is called time domain OCT. So it has an interferometric setup. So we have a light source, and as I was mentioning, because you can't measure the time of flight because it's too fast, to be able to measure the signal detected back from the sample and to reconstruct an image, we make an interferometric measurement. So we use a low coherence light source, and the light is split between two paths. One goes to the sample, and one goes to a reference arm, which has a mirror. So you get a reflection back from the reference arm, and you scan that mirror in the reference arm at different positions so that you can probe different depths inside of the sample. Because the light source we use has a low temporal coherence, so that we only detect interference when the difference in path lengths between those two arms is below the coherence length of the source. So we get interference and then detected, and the output is the sum of the two fields, the reference field and the sample field. So it's proportional to the square of the total field. So as we scan the path in the mirror, we get an interference fringe. And because of the short coherence length of the light source, that is localized to different positions in the sample. So this is the principle of time domain OCT, which is how OCT was initially introduced over 30 years ago now. And then since then, the main way that OCT is used is in Fourier domain OCT, which is kind of illustrated here. And what the concept here is that you have kind of the same basic blocks, but you're no longer scanning the mirror in the reference arm. So it has a fixed position. And what happens is that different layers in the sample cause different path length differences between sample and reference arm. And that has an impact on different frequency of modulation of the interference spectrum. So, for different differences between that path length, you get a different modulation of the spectrum. So in the Fourier domain OCT, we acquire the spectrum of the interference. And with the Fourier transform, we get the depth profile. And the advantage is that, at a single point, really you get the entire depth profile without any moving parts. And then you can scan over, kind of raster scan in the 2D, and you build a 3D image. So what is, I think, really unique and advantageous in OCT is that axial and lateral resolutions are decoupled, so you can kind of control those separately and tailor them for different applications. So the axial resolution is inversely dependent on the bandwidth, delta lambda, so here on the left. So the broader your bandwidth of the light source, the better the axial resolution. And the lateral resolution as an imaging systems depends on the numerical aperture of the system and central wavelength. So in imaging systems, there's always a trade-off between the lateral resolution and depth of focus. So if you increase your numerical aperture of the beam of light, you get a better, tighter spot, better lateral resolution at the expense of depth of focus that becomes more shallow, as shown here with the green situation. Typically in OCT, we use a lower numerical aperture because that allows you to get a depth of focus that covers the entire depth of the sample. And that's at the expense of the lateral resolution that's typically in the tens of micrometers. When in microscopy, you typically have the green situation here with the higher NA, but limited depth of focus. So here's kind of the same concept shown as a chart. So I have the lateral resolution on the left and the depth of focus on the right, both as function of numerical aperture. And you can see that ideally you would want to be in these kind of regions. So you have a low resolution and a large depth of focus, but they don't overlap. So that's kind of the challenge in achieving both in the same system. So an approach to break this lateral resolution limit for OCT is to increase the numerical aperture, and that reduces your depth of focus, but you can focus at different depths and acquire multiple volumes and then fuse them together to obtain a kind of high resolution, full depth image. And that's the approach that's called Gabor domain optical coherence microscopy. So we have a relatively large numerical aperture, and we acquire volume, and then we refocus deeper inside of the sample. Here is shown for three different positions. And then we extract the portion that's in focus. So showing here is kind of the cross section, and you can see that the bright portion is kind of moving down in the image, and that shows where you're focused, so you get the better contrast. So then we can extract that portion from each image and fuse them together in a single image, single volume that maintains the high lateral resolution throughout. And so that's how we can achieve a 2-micron resolution, both laterally with acquiring multiple volumes, and axially based on the light source. And this is an example of a finger, a human finger, and some basal cells in the finger. So with OCX, then we can integrate both OCT and GDOCM in a single instrument. And so that gives you this kind of capability. So this is a human cornea. So with OCT, we can image it over a large field of view of 10 millimeters. This gives you basic morphological information. But if we switch to the microscopy, we can get down to the individual cells that line the posterior surface of the cornea. And the advantage of having both in the OCX as a single instrument is there's no sample repositioning, but you can characterize on different scales the same sample. So OCX is a kind of new class of imaging that combines these two capabilities in a single instrument. And we produce volumetric images. There's a fruit fly with 2-micron resolution in 3D. And the field is large compared to microscopy. So this results in non-invasive structural assessment of tissue, kind of producing the optical biopsy capability. We have done comparisons with the GDOCM and traditional histopathology, for example, on skin cancer. And since these data sets are very dense of information, it's very advantageous to combine them with machine learning to automatically extract parameters of interest. Here's an example that we'll see later on as well on cornea cell counting done with the machine learning. And finally, there's also the potential to integrate the structural imaging capability of OCX with functional assessment. And I will show later on an example of extracting the vascular information from the OCT images using OCTA, a technique that's called optical coherence tomography angiography. So here is a picture of the instrument from where we started. There's been many iterations now. It's a very compact desktop format system. Everything is shown here. There's no extra computer. Everything is in the box. So we have a microscope. Right, in this case, it was mounted vertically for imaging a mouse eye. And so the system operates at in the near infrared at around 850 nanometers. So that gives us an axial resolution of 2.2 micron. And the lateral resolution depends on the optical system. So it depends on the numerical aperture. And we have two modalities. We have the OCT modality and the OCM modality. So the OCT modality has lateral resolution of 13.5 micron. The OCM modality has a resolution of 2.8 micron. And they have also different fields of view. So the OCT is at 10 millimeter. The OCM is up to 3 millimeter. And we also have a video camera that's integrated that shows the alignment of the sample. So here is kind of a side-by-side image of a retinal organoid. So image with the OCT on the left and with the OCM on the right. And I like to show this because and then we're kind of scrolling down within the sample. And you can really appreciate the advantage of the high resolution in resolving the features of this organoid. Here's some still images of that same sample. And really the advantage is that you're able to non-invasively evaluate a sample and then look at different cross sections. You can perform measurements and quantify the morphology of the sample. So in terms of applications, we talked over lunch about the breadth of applications for OCT, starting with the retinal and how it has revolutionized retinal imaging. In our case with OCX, it's really adding to OCT that cellular capability. So we've done a study on imaging the cervix. So these three applications are the ones I'll be showing the remainder of the talk. So there's cervical imaging, there's the vasculature in skin, and the corneal imaging, in particular looking at endothelial cells. So this study we've done in collaboration with the U of R, University of Rochester Medical Center. And the background is that there's a cervical cancer screening program and HPV vaccination program that have had a significant impact on the incidence of cervical cancer. However, in the United States, cervical cancer still affects 13,000 women per year, with approximately 4,000 cancer-related deaths. And so patients that present an abnormal cervical cancer screening are referred to colposcopy for diagnosis and management. So colposcopy uses a binocular microscope to visualize the cervix and identify abnormal areas. And the abnormal areas are biopsied and sent for histopathology evaluation. The histologic evaluation of the punch biopsy is the gold standard and for identifying cervical intraepithelial neoplasia. So the biopsy result determines whether the patient needs treatment by excision of the transformation zone with a procedure that's called LEAP, loop electrosurgical excision procedure. So the challenge in this current screening process is that it suffers from limitations such as poor or decreasing volume of colposcopy. So most providers are doing now less than 50 cases per year. So the skill of the providers is declining, and there is poor reproducibility. The sensitivity is also low, as this is a biopsy. There's studies that show that taking a single biopsy targeting the worst appearing lesion may miss up to a third of precancerous lesions. So achieving an acceptable level of sensitivity requires taking between 2 and 4 biopsies. So the question we tried to address with the study was, is there a role for advanced technologies to perform optical biopsy in this context? So in this study, our purpose was to establish whether the GDOCM can distinguish between cervical epithelium and stroma and distinguish cervical intraepithelial neoplasia, and compare this with the gold standard of histopathology. So we conducted a prospective ex vivo pilot study on 5 women undergoing LEAP. And so this was done in the surgical center at URMC and right next to where the LEAP procedure was going on. We had the specimens right after they were excised. They were tagged at 12 o'clock, and we only had 20 minutes to perform the imaging experiment, because then the sample had to undergo the regular preparation and the routine histopathology and processing and review. So here's a look at what the cross-sectional image looks like. We call this cross-sectional image a B scan. So this was the B scan of one of the specimens. And this cross-sectional view is also the typical presentation that you have with histopathology. So in the GDOCM images, we achieved cellular resolution, so we could resolve individual epithelial cells. We could identify the junction between epithelium and stroma. And we could visualize hyperkeratosis, which is a non-specific feature seen in dysplasia. And this was noted in about 62% of the scans. So other findings include papillary structures and stromal vessels. So the grades of cervical intraepithelial neoplasia are determined by assessing the density changes over the epithelial layer. So it's shown here from going from normal on the left to CIN 2 and 3. And here are some examples of the corresponding GDOCM images that were graded by a pathologist as CIN 1 on the left and CIN 3 on the right. So in this comparison, we have images that were graded as CIN 3 for the same sample, the corresponding GDOCM on the left, which presented an increased density, leading to the CIN 3 grading. So this was a preliminary study that demonstrated new capabilities with the 3D imaging that can achieve this assessment non-invasively, and it was validated against the gold standard of histology. So next steps for this applications are to integrate the imaging capability with the surgical workflow, and possibly automate the grading of the images. So the next application I wanted to discuss is vascular assessment of skin with OCT angiography. So this is a functional imaging capability that can enhance the structural imaging of OCT and GDOCM. So blood vessels and tissue support critical metabolism function by exchanging nutrients and oxygen with cells. So OCTA visualizes the flow of blood cells by acquiring a repeated scan over time on the same location, and high-pass filtering in post-processing. So you can extract the large variations in repeated scans that are caused by the moving blood cells. And so this is a label-free technique. The challenges, however, especially when we're dealing with OCM, so the microscopy, high-resolution OCT, are motion artifacts. This is obviously a technique that needs to be done in vivo because the blood needs to be flowing. So you're definitely dealing with the bulk tissue motion as well. And moreover, there's additional challenges because when you have a high resolution imaging method, you also have high sampling, which takes a longer acquisition time as well. And so there's many approaches described for performing OCTA, but we've proposed a novel approach that has the advantage of reducing the motion artifacts, which was really important within the context of high resolution imaging. So we did this by separately modeling the motion in the image processing. And this is again a factor analysis based approach. So the processing pipeline is described in this diagram, where we have repeated scans that are linearly fitted as a combination of unique terms and common terms. So we model the B-frame correlation as two independent correlations. So practically, this was done through iterated factor analysis that maximizes the log likelihood probability. So next, we back transform the high order common terms into space domain, and then sum them and do a log compression. So essentially, OCTA is a high-dimensional two-class classification task to differentiate the static structure and blood flow, in which the linear fitting itself may not well approximate the classifier due to nonlinear correlations with respect to the time interval, flow speed, particle concentration, and so on. So to deal with this issue, we added a subnetwork through a list of nonlinear operations that include summating the original features, the original frames, log compression, median pooling, rescaling, calculation of power ratio, and importantly, transformation by a reverse sigmoid function. So the final OCTA map was obtained through a pixel-wise multiplication between the blood flow image from linear operations and the map of non-linear operation. So we named this method ultra-high resolution factor angiography. And so this is the image acquisition and analysis workflow using the OCX instrument, in particular with the OCM, GD-OCM capability. So we acquire an image with 2-micron resolution, and we repeated the imaging 5 times at the same location to get the dynamic functional information. Next, we applied volume registration. We've developed a motion correction algorithm. So we've in this frame align tool. Next, we perform the OCTA analysis. And finally, we can visualize the results in the viewer 4D software. So here as an example of a densely sampled GD-OCM data set of the nail fold of the human finger. And there are these are the corresponding views of the en face. So it's parallel to the surface of the sample, and processed in 2 different ways. The top is the conventional OCTA processing, and the bottom is the factor analysis. And you can appreciate the reduction in motion artifacts that allow us to better resolve those U-shaped the capillaries in loops in the bottom approach. And according to the literature, the density of these capillary loops is between 9 and 12 per millimeter, which agrees with our results. So here we have another example of the OCTA on the left for a normal finger, and on the right there's a knife wound, and you can so at the bottom we have the structural OCM image, and then the OCTA with a traditional approach, and then with a factor analysis approach. And you can see with our novel approach, there's an improvement in the contrast to noise ratio compared to the conventional. And additionally, the factor analysis approach reduced the motion artifacts by nearly 50% compared to the conventional when evaluated on local differences. In these in the wound case, you could see some interstitial fluid and biofilm that gets accumulated in the wound. So we developed this novel motion insensitive OCTA approach that's based on factor analysis that can efficiently reduce motion artifacts that are highly significant in high-resolution OCTA imaging. And motion artifacts were reduced by nearly 50% on average, evaluated on local differences compared to the conventional approach. So let's move on to the application of corneal imaging for evaluation of transplant tissue at eye banks. And So at iBanks are tasked with evaluating tissues that are from donors prior to assigning them for a transplant. And the tissues are stored in these viewing chambers that are shown in the picture. So they're kept in an isolated environment so that the tissue is not exposed to contamination. And so we've designed our system so that we can image through that container without having to open it. So today, iBanks use a number of different instruments to evaluate the tissue. And we proposed our instrument as a one-stop solution for iBanks. So we have the capability on the left of the OCT image over a 10 mm field of view. So this can be used to perform the thickness measurement that iBanks perform. And on the right, we have the OCM capability that has the cellular resolution throughout the thickness of the cornea. So you have the movie is kind of the en-face scrolling down through the cornea. So once you have the OCM image, you can it's a 3D image, so you can look at different cross sections and evaluate the different layers of the cornea. And in particular, looking at the en-face view, we can appreciate epithelial cells, keratocytes, and the stroma, and the endothelial cells, which are critically important as a metric for the health of the cornea. In fact, there are guidelines for iBanks of cell density for the endothelial cells that allow them to accept tissue for transplant or not. So the way they perform this measurement today is using a specular microscope to image the endothelial cells. And specular microscope produces a single image of the endothelium over a small field of view, and the technicians currently count the cells manually, and then it's extrapolated by the machine to produce a cell density measurement. So our goal was to automate and improve this process. So from the 3D image of the cornea, we've implemented an automated segmentation so that automatically the endothelium is segmented. Now, because the cornea is naturally curved, we implemented a numerical flattening so that we can present those cells in a single plane. And then to perform the automated counting, we had to build a training set for our convolutional neural network. So this was done by some students at U of R. They did a manual cell counting on the images. So they used 6 corneas, and for each cornea, they imaged 5 different locations and repeated 6 measurements per location, so a total of 180 data sets. So these are the 6 corneas. And from these images, the manual cell counting was performed. So this is the results of the manual counting for the 6 corneas, and at the 5 different locations, and the error bars show the repetition per location. And so we the repeatability over the different areas was less than 2%. So once we had this manual counting, we could use this as the training for an automated approach. And we've developed a cell counting method to automatically detect and count the endothelial cells, and using the manual cell counting as the gold truth. So we trained a convolutional neural network to automatically segment the OCM images of the endothelium. So that means to automatically detect the cells and the cell boundaries. And the training was done on 150 images resulting from the 5 manually traced corneas, and tested on a total of 30 images from the remaining corneas. And this procedure was repeated 6 times in a leave one out cross-validation strategy, leaving out a different cornea for testing each time. So here's an example of the results. So we have the image on the left, and on the right we have the segmentation of the cells. So we have the red for the manual only, and the blue is the automated only, and white is common. So there's great potential for the automated approach to do a better job than the manual counting. And this really addresses a key limitation at the iBanks today, because as I mentioned, the technicians start from an image that's similar to this one on the left, smaller field, but similar, and they manually click on each cell to indicate which one has to be counted, but they have a tendency to pick the ones that look better. So there's an inherent bias in the process. So the goal is to move to a fully automated, unbiased approach. So here, so then once we've trained the network, then we image some more corneas and tested. Oh no, this is actually, yeah. This is, yeah, so this is the comparison between the manual and the automated cell counts on 6 additional corneas. And the average difference in the endothelial cell density was 0.3%, and with a Pearson correlation with an R squared of 0.94, and these results were statistically significant. So to validate this, additionally, we performed imaging of 10 samples on with the GDOCM plus the automated neural network approach, and compared it with the traditional specular microscopy evaluation done at the iBank on 10 corneal samples. So here are the results. So they're manually on the specular microscope and automated on the OCM. And we had an average difference in the cell density of 3.7%, and the correlation had an R square of 0.91. So here's an example of the images. So the left is the specular microscopy, and on the right is the GDOCM. So for this application, we've demonstrated a new capability of being able to image the corneal tissue in 3D with cellular resolution. And we validated against the gold standard of specular microscopy, which is limited to the endothelium only. So having the automated cell count is really important for, as I was saying, for the unbiased assessment of the tissue. So it's really important to fit in the workflow of iBanks. So here are some examples within cornea now for in vivo imaging of mouse cornea. On the left, the OCT view of 10 millimeters, and on the right, the microscopy. So we're able to resolve cells in the epithelium on the left, endothelium, and corneal nerves as well. So within the context of corneal imaging, combining OCX and machine learning automated cell counting was shown to produce results that are not statistically significantly different from the current practice of manual counting from the specular microscopy. And in the mice imaging, we evaluated nerves, nerve fiber lengths, and branching, and compared 2 different groups, normal mice and diabetic mice, and we were able to observe a reduction in nerve fiber length greater than 30% in the diabetic mice, which was consistent with the literature for diabetic neuropathy. So in summary, the optical biopsy capability with the dual modality OCX enables non-invasive 3D in vivo imaging of tissue with the cellular resolution over a wide field. And integration of image processing and machine learning and other advanced technique can enrich the 3D structural imaging capabilities of OCX. And we can extract dynamic properties of tissue with a factor analysis approach for extracting OCT angiography information and reconstructing the vasculature signature of tissue. All this work has been contributed by a number of individuals over the years, from scientific to technological, commercial, medical, and everything else. So a lot of them are shown here in no particular order. And with that, I'd be happy to take questions.
Any questions? The audience? No, facilitate the Q&A.
Thanks for the talk. So since the nature of interaction with these tissues will change, every tissue has different scattering, I guess. How
Yes.
do you use one source to image everything?
Yes, that's a very good question. And there are a few options within OCT based on the biological windows of tissue. So there's and availability of components and light sources and all of that. So there's kind of 3 good windows, which is where we are around 850 nanometers, then around 1 micron and 1.3 micron. So the choices you have to make are based on penetration inside the tissue. So the longer wavelength gets you better penetration, but the drawback is you lose in the lateral resolution. So in our case, because we're really focused on the best lateral resolution you can achieve, we're at the shorter wavelength range. But there's a lot of interest in the longer wavelength, especially for skin and highly scattering materials. The fact also the cornea is not highly scattering, so having shorter wavelength is perfectly fine. We can image through the entire cornea. But those are kind of the choices you have to make in working with OCT.
All the 3 applications.
These are all at 850. Yes, same source. Yeah. And so there's an imaging depth you can achieve with the interferometer, but the tissue is always going to limit that. So in our system, we can get up to 3 millimeter penetration, imaging depth, but the actual penetration you get depends on the scattering of the tissue. So it'll always be less than that if you're in a highly scattering tissue.
Yeah. I guess I just had a general question. If you go back to slides somewhere between, if you start at 45 and go backwards, it was the counting. I just want to understand that graphic.
Oh yeah. Which
This was a summary phone. I think that sets it up. So maybe it was more forward. It was the result where you're actually showing. Maybe it's the next one. Right.
This one?
One back.
Oh, okay.
Yeah. So I was just trying to understand this.
Yeah.
I could ask this question offline too, but since we have a couple minutes.
Yep.
So the deal here was when you say manual only, right?
Yes.
And you have common, so that's both.
Yep.
And it looks like automated using ML actually used CNN.
Yes.
Yeah. So that gives you, looks like more than manual, but what's
Yeah. What is the ground truth?
is it that there's manual means you're supposed to go through and try to label as many of those as possible, but could it?
Yeah, so the next step, we should go back and review the manual again and see, okay, is this actually one that we missed?
Yeah,
And that's
the ones
blue.
that are, the ones that are cyan, that's not manual, right?
No,
Is
that
it
was,
because somebody said, I don't
yeah,
think
that's
the
well,
it's
no.
subjective, or what does that mean? I guess that's my question.
Well, the manual counting was done first, and that we used
Yeah.
as kind of the ground truth, but looking at these results, we want to go back and see, well, maybe the manual wasn't as accurate as it could have been. Maybe they missed some.
So, me, I still don't get it. So where it says automated only, it's the cyan color, well, that's
Yes.
a big chunk over on the far left.
Yep.
So there's no manual count, so manual counting wasn't performed in that region? Is that?
Right, so
That's
the manual was done by a person that wasn't able, or decided not to label, annotate that
Right, so
region,
that
yeah.
gets to the root of my question,
Yeah.
why that is. Is it because they're like, I don't know what this is?
Maybe they
Look at
weren't
their
sure.
cells.
Yeah, yeah,
But the algorithm thinks they're cells.
right, right.
Okay, all
Yeah.
right.
So that's why it's
That's-
exciting.
then
Yeah.
that's why it's encouraging, yeah.
Mhm.
And then the follow-up to that is, so in the image that you have on the left, right? I mean, just being silly here, I could take that into Photoshop and stretch it, contrast, enhance, and play games.
Sure,
all that kind of
sure.
image processing.
Yes.
What kind of ramification does playing those games
Mhm,
have on the machine learning? Does it actually
Yeah.
have to represent the training that you did, or if you perturb the input, you're in trouble? How much slop do you have in, you know what I'm asking?
Yeah.
Yeah.
I mean, I believe we just work with the raw images. I don't know. I'm not entirely sure what the manual, like, if they did any of that enhancement before doing the manual.
Yeah, in other words, if the
Yeah.
image looked different
Yeah.
and it's not different contrast
Yeah.
wise from the training,
Yeah.
does it say, I don't even know what I'm looking at and it performs poorly? I was wondering how much latitude you have in that. But
Yeah, I'm not sure, but yeah, that's why I was mentioning earlier in the lunch is really the amount of data is our limiting factor. The more data you have, the more you can really probe how robust the approach is. And this is a new instrument, so all the data is just getting collected.
But initially out of the gate, obviously, this is encouraging because you have more cyan than manual, so.
Yeah.
They both have
Yeah.
plus the common stuff, so.
Yep.
Anybody else? Yeah.
So in the slide where you're showing the depth, varying the depth to get the layer, did you do any kind of three-dimensional point spread function estimation kind of stuff to kind of tailor to get better resolution? Is that something which is helpful in this?
I'm not sure. I think you mean when we refocus at different depths.
Yes.
And so
So there is the depth of focus is limited, right?
yes.
But if you put the point spread function across the z-axis, will that information be helpful in this imaging system?
Yeah, I mean, we can measure the point spread function independently through just moving we could put a reflector in the sample arm and scan it through. We don't use that information, but that could be something that could improve. It could be used. Yeah, we don't use it currently.
Maybe I have a follow-up to that.
Yeah.
So when you're going in this direction and so if you focus, image, focus, image,
Yep.
focus, image, right? You can't, or maybe you can, it's part of my question, you can't do that absolutely theoretically continuously. Do you have to interpolate? So you're at sort of getting the root of my question. If I had some volume, but I only can sample at 5 positions,
Yeah.
there's areas where I'm not imaging.
Yeah.
But when you made those movies, you're moving through, you have to do something about the data between.
Yeah.
Is that a thing? Do you have to interpolate, or it's so fine that you don't care, or what's?
Yeah,
what can you look at?
so the depth of focus is about, it varies, it could vary a little bit based on the sample, but it's between 50 and 100 micron.
Okay,
Our
so
pixel is
then do you
less-
end up overlapping so that you don't have, because the depth of focus ends up being kind of, some fat. into it. So
yeah, we
just
do some kind of blend, like we kind of blend we call it fusing, but yeah, we blend
So
the
that's-
regions in between. But definitely choosing how many different positions to use is sample dependent.
Yeah.
Yeah.
Yeah, so that answers
Yeah,
the question.
yeah.
Any last questions? All right, let's thank our speaker. Thank you. And 3 o'clock, I'll see the